There's about 56 different questions that we're trying to answer at our research farm, and then deliver that information to the farming community. We know improving the quality of the soil will help farmers build resiliency on their farm. When the weather is as erratic as it is, this is the only buffer that you have against that weather.
Hi, my name is James from Barn to Door. We help thousands of farmers across the country increase sales and save time online. Join me for today's session, 2021 Trends Every Farmer Needs to Know, where we're going to cover the tips and tactics that your farm can employ to build a thriving business online.
Hey, hi guys. My name is Olga Redding, and I'm with Iowa Organic Association as well as Midwest Organic Pork Conference. And we are so thrilled you decided to be part of this amazing, growing, stronger collaborative conference. And we are one of the partners and are happy to present one of our amazing speakers, Diane DeWitte. I hope I'm saying that right. And so the workshop is managing swine intestinal parasites continues to be an obstacle for organic pig farmers because of the lack of organically approved options for controlling them. This workshop explores manure and pasture management strategies that help reduce parasite contamination and transmission in organic pork production. And a little bit about Diane. Diane is with University of Minnesota Extensions. She serves as an extension educator who provides research-based information on swine production, health, reproduction, and biosecurity to pig producers. In this role, she also focuses on production practices in small farm, organic, and niche swine production. She currently serves as part of the university's research team investigating the prevalence of parasites and their control in organically raised pigs. Diane, welcome to the conference. Thank you for being a speaker on behalf of Midwest Organic Pork Conference. We are so delighted to have you here.
Thanks so much, Olga. I sure appreciate that. This was, I've always, we've had one organic pork conference in Waterloo, and wanted to have the second one last year in Dubuque, and unfortunately weren't able to. But the work goes on, and I'm sure tickled to be able to give this presentation here in this venue and to let folks know what it is we've done and what we are continuing to do related to parasite control in organically raised pigs. The first thing I'm going to do is share my screen, and let's see here. Share this. It should be coming, right?
Yes, we are seeing the presentation.
Okay, awesome. And let me go to the slideshow. And then I need to swap this. How's that look?
Yes, perfect. Thank you.
Okay.
Now it looks great, Diane.
Yeah. So this has been a collaborative effort. Of course, we always enjoy collaborating. And actually, I think that the origins of the collaboration began with the Moses Conference a few years ago, when my colleague and the principal investigator from the University of Minnesota end of this, Dr. Yushi Lee, met Alex Hernandez from Kutztown University in Pennsylvania, and also Rick Carr, who is with Rodale Institute in Kutztown, Pennsylvania. And they got to talking about pasture raising pigs organically, and in pasture settings and outdoor settings, and the parasite issues that we always see. And so they began a collaboration, and they were able to secure a USDA grant that was part of a USDA Organic Transition Program. And so they put together the proposal for the grant, and were fortunate enough to be awarded the grant. And so set about doing this project. And at the time, and at the beginning, we've had 4 objectives. The first being to actually research and measure how the prevalence of parasites in organically raised and outdoor raised pigs. Of course, we know that it's there, but it never had really been quantified. And the ability to have a grant that supported this research is really important, just so that we know. We know it, but we want to be able to have measured it and know what kind of numbers we're looking at. The second objective was then to try to find out some ways to remediate parasites in a way that doesn't use synthetic parasite control. And so the second objective was to determine the effectiveness of manure composting in remediating and eliminating parasites in organic pigs. And then the third objective was to assess bio-fumigation as a parasite management approach. And when we get further into the discussion here, I'll talk a little bit more about bio-fumigation. And then our fourth objective was to determine the effects of grazing bio-fumigated pastures by organic pigs. And that's an element of the research that's still ongoing. So our first objective was to evaluate parasite prevalence on organic pig farms. And so because we were working with partners in Pennsylvania, and we're here in the upper Midwest, Dr. Lee is located at Morris, Minnesota, which is central Minnesota, and I'm located in southern Minnesota. But we were looking at parasites on organic farms in Minnesota, Iowa, Wisconsin, and Pennsylvania. And as it happened, on the 9 farms that with whom we were working, they represented about 30% of the national organic pig farms, and were sizable organic pig farms, because their national organic pork sales, their organic pork sales represented about 60% of the national organic pork sales. So we were really happy to have participation by a group of farms who have an impact as far as organic pig rearing and organic pork sales in the country. And so we did on-farm data collection. As I mentioned, we had 9 farms with whom we worked in the 4 states, Minnesota, Iowa, Wisconsin, Pennsylvania. And so the criteria for the farms who participated were that they had to be organic. They didn't have to be certified organic, although some of them were. They needed to be what could be termed alternative or niche, or at the very least have outdoor access. The pigs had to be able to get out, be reared indoors and outdoors. We wanted to see bedded flooring. We didn't have any producers in the study with slatted floors, nor what you would consider confinement type operations. No synthetic dewormers were used, and these farms needed to have raised pigs in this manner for at least 3 years. So that's how we started, and we were collecting samples. We were trying to understand and quantify the prevalence of parasites in these pigs. So we were collecting samples. Of course, you normally think of feces, and so we were picking up fresh specimens on the ground in different locations at each operation. And then we were separating the collections that we were making by the stage of production. We had the breeders, which would be sows or boars, during gestation, and all parodies of sows. And then we were collecting and segregating the samples from growing pigs, and those were pigs that were less than 5 months old. And then the finishing pigs were 5 months old to market weight. And so again, we had 3 categories of pigs in the samples that we were collecting, and we separated the feces according to which situation they were. We also were collecting bedding samples, and so we went into the barns and collected the straw or the shavings or whatever bedding that there was. We were collecting samples looking for parasites in the bedding, and then we collected soil samples from the outside area on these farms as well. So we felt like we were covering the gamut pretty well when we're collecting actual feces and then bedding and soil samples. And so again, we had 9 farms with whom we worked, and we actually went to these farms, and this was in June and July of 2019 when we did this work, and visited the farms. There were 9 farms that we visited. I'm going to mention 5 here just because they were specific styles that we were looking at, and our collaborators in Pennsylvania did the collection out there. The Minnesota crew did the collections in Minnesota, Iowa, and Wisconsin. But one of the farms with whom we worked was a certified organic farm in Iowa, and it was a GAP 3 certified farm. And if you're just for a little information, the GAP certified is a program. GAP stands for Global Animal Partnership, and there are 5 different levels of GAP certifications ranging from 1 to 5, and the GAP 3 certification is that they're raised without antibiotics or added hormones or any animal byproducts. And with all GAP certification, they're audited every 15 months. And in GAP 3 certification, the minimum weaning age would be 42 days. So they were weaned at 6 weeks old or older, and there isn't any tail docking or needle teeth clipping or detusking of the boars. So GAP 3 is kind of a common certification for pigs raised in this style. These pigs on this particular farm in Iowa were marketed to Organic Prairie, which is one of our conference sponsors. So we're glad to see Organic Prairie continuing to support the work. This farm in Iowa actually marketed 350 pigs per year, and they were raised in barns that had outdoor access. And so here you can see Dr. Lee is, this was during the sample collection visit. And this farm was actually a 35 sow breeding herd. They had 4 boars on the premises, and they farrowed both winter and summer. They pen farrowed in the winter inside, and then they pasture farrowed in the summer. Another farm, this was our farm, one of our collaborators out in Pennsylvania, and they were certified organic and were GAP 3 certified, and they did direct marketing of their pigs, and they marketed about 60 to 80 pigs per year. They had a more modest breeding herd of, at the time, 4 sows and 2 boars, but a very nice hoop structure that they worked out of. And each of these pens inside had outside access to pasture. They were runs that the pigs were separated, but they each had a sizable pasture run outside their pen. And all of the pigs in the group were raised within this same hoop barn. An additional farm that we visited, a certified organic farm in Minnesota, also GAP 3 certified, and they marketed all their pigs to Organic Prairie, and at that time it was 500 pigs per year. So they had a 40 sow breeding herd, and they had several boars. These were barns that had outdoor access, solid flooring, bedded floor inside, and then outside pens. They didn't have access to pasture. And then the growing pigs and the sows and boars were in separate barns. An additional setting that with whom we worked was a not certified, not organic certified farm in Wisconsin. This producer purchased feeder pigs and weaned pigs, and most of them were Berkshire extraction of some kind. Either they were purebred Berkshires or they were a Berkshire cross. And at the time that we were there, these pigs that were actually out on pasture were all from the same source. He did have some smaller pigs that he was sort of backgrounding in the bedded barns that were from multiple sources, but the actual pigs that we went out into the pasture and looked at were all Berkshires, and pretty much these were all purebred Berkshires as well, from one source. But he direct marketed about 100 pigs per year, and he marketed them through various venues, either farmers market or sold from the farm. But he would purchase feeder pigs. He got them smaller in the spring, and then in the fall, generally he was purchasing them more 70 or 80 pounds, and he fed them a little bit differently too, depending on the season. The pigs that he purchased in the fall were consuming more apples and pumpkins and that kind of produce. In the spring, we're eating a diet that they fed them out in the pasture. And then an additional certified organic farm that we visited in Wisconsin, also this producer direct marketed these pigs. And in 2019, he was marketing about 20 pigs. He had a variable sow herd size. And at the time we were there, it was between 7 and 12 sows. And he had at that time, he had a purebred Berkshire boar also that was with the sows, and they were on pasture during the summer. And the market hogs then were in barns that had outdoor access. And you can see that this was, it was sort of a moderate pasture access. And so in trying to decide and quantify the types of and the amount of parasites that were present in these outdoor pigs. We found that we were investigating 3 specific species of parasites. The most common one by far that we see in pigs anywhere is the Ascaris suum, which are the large roundworms. And this just shows, this slide shows, you know, what we're looking at. And it's not uncommon to find these actual worms in the pasture, you know, in the feces that are being excreted by the pigs. The life cycle of the Ascaris is that eggs are shed, adult worms are in the gastrointestinal tract of the pig, laying eggs. Eggs are shed through the feces, and so the eggs are found in the pasture on the ground, and the pigs will consume them as they're grazing in the pasture. They'll pick up eggs, and with Ascaris, they get into their GI tract and they begin to develop. And as they develop, then larvae will move through in the GI tract into the liver. And that's why we saw in this previous slide an infected liver. You can see that it's been affected by the Ascaris. And then from the liver, then they will proceed through the bloodstream into the lungs, and they do some developing in the lungs. And this is why a lot of times with Ascaris infections, you'll hear coughing because the lungs are affected. And as the larvae develop and get bigger, then they're kind of coughed up and swallowed back into the digestive tract, and they grow into adults, and then again lay eggs, and that's the cycle. The thing about Ascaris eggs is they can live for a long time in the environment, and so it's not uncommon for Ascaris eggs to be present in the pasture for years, 4, 5, 6 years. The second parasite that we were looking at were the Trichuris suis, and those are commonly known as whipworms. And you can see here what the egg looks like and what the adult worm looks like. And the life cycle of the Trichuris is very similar. Doesn't have the liver and lung and back into the gut kind of movement that the Ascaris does, but with Trichuris, the eggs are laid and shed through the feces, and then they're present in the pasture, and the eggs are picked up by the animal and consumed, and then the actual Trichuris develops in the digestive tract, lays eggs, and they're shed through the feces. And then the third one that we were looking at were Osphagastumum, and they're more of a nodular worm, and they develop again in the digestive tract, and then eggs are shed, and they don't live as long in the pasture as the Ascaris and Trichuris, but eggs are shed, picked up by the pig, and the life cycle occurs within the digestive tract. So we were collecting these samples, and I mentioned earlier we were collecting feces and also bedding and soil samples from these farms that we visited in the summer. And this was, summertime when the pigs were outside or out on pasture, and we were picking up these samples. And what we did, as soon as we picked them up that day, we packed them in a shipping container with dry ice and shipped them overnight to the biology lab at Kutztown University. And Dr. Alex Hernandez was there. He's one of our collaborators. And they used the McMaster technique there to analyze these samples for parasite eggs. And here you can see one of the summer lab technicians working with Dr. Hernandez. And there's Dr. Hernandez himself at the microscope. And it turned out to be a very it worked out very well because we were able to get the samples delivered to the lab in Pennsylvania overnight, and they were still in good condition when they got there, and they always knew that they were coming, you know, so they were able to process them right away, and we were able to get really good results right away from the lab at Kutztown University. And as you may recall, the Rodale Institute is also there at Kutztown in Pennsylvania, and of course, they collected the samples from their pigs there at Rodale, but they also worked with some producers in Pennsylvania to get some additional samples beyond the ones that we were collecting in Wisconsin, Iowa, and Minnesota. So here's just a little bit of a look. The team that traveled and did the sampling that I worked with, the Minnesota team, included Yushi Lee, the primary investigator on the project from Minnesota, and then a graduate student that was working with Dr. Lee, and then a summer assistant that was working there with Dr. Lee at the Research and Outreach Center at Morris, Minnesota. And then I went along too. And so I was had a kind of a different role. My role is actually rolling out now as someone with Extension who's actually helping disseminate the information. But at that time, I was sort of the videographer and photographer of the project. And here we are. This was at one of the at the organic farm in Minnesota, in western Minnesota. The team was visiting with the producer there, and this was the organic farm where they were raised in a barn and outside pens, but they actually didn't have pasture access. So in total, we were able to collect 243 fecal samples from the 9 farms, and we were quantifying the parasite prevalence and the load. So with the prevalence, we were looking at the percent of pigs infected by production stage and by farm, and then we were able to identify what percent of the farm was infected and by production stage. And then we were also looking at the load. And we were measuring the load by parasite eggs per gram. You can see EPG, and in some further tables that we'll show, that's what eggs per gram is what we're looking at. And if we had 0, that we considered that there weren't any, or in further data that we'll look at a little bit later, if the EPG is 0, we will consider them neutralized. But we were looking at the fecal egg count and then quantifying it average for the farm by production stage and by the farm. So we were comparing the farm within itself and also the 9 farms together. So what we saw as far as the parasite prevalence on the 9 farms, we saw that 8 of the 9 farms were infected with Ascaris, 89%, and the prevalence on each farm ranged from not too bad, 9% to 78%. With Trichuris, we saw that 44% of the farms were infected, and the prevalence of Trichuris on the farms, now remember those were the whipworms, we saw 22 to 54% prevalence on the farms that were infected. And then Oxyphagostomum were the more nodular worms. 67% of the farms were infected. So we found Oxyphagostomum on 6 of the 9 farms, and the prevalence ranged at anywhere from 6% to 100%. So as we break it down, you can see Oxyphagostomum is the green bar, and Ascaris is the blue bar, and Trichuris is the red bar. And so we went everywhere from, we saw parasites on every farm, and how many and what we saw varied, widely. On farm 9, we had only Ostracismum, and on the rest of the farms, we had a combination of the other two. So when we look at the prevalence of 1, 2, or 3 parasite species by production stage, we saw that 72% of the pigs were affected. 57% were affected by one parasite species of a kind or another. 13% were affected by 2, and then only 2% of the pigs were affected by all 3 parasites. And here you can see across all stages, the breeders tended to have 3 species. There were some that had 3 species. Oh, I didn't say that right. They either were not infected or they had 1 species. Blue is 1 species, white is 2 species, and black is 3 species. So with the growers and the finishers, they had all 3 present. And across all stages, we saw some prevalence of the species. And so in different production stages, we saw more of the breeders had Ostracismum compared to 8% of the growers. Now remember, the growers were the ones 5 months old or younger, and the finishers were 5 months old to market weight. And so not so many Ostracismum in the younger pigs, but we saw them in the adult breeding animals, and we saw them in the finishers. With Ascaris, which were the roundworms, we saw more of them in the growers and the finishers. 64% of the growers and 64% of the finishers were infected with the roundworms. And with Trichuris, which were the whipworms, again, we saw more of them in the growers and the finishers, and not so many in the breeders. And here, this shows you a bar graph of what I just mentioned, the prevalence at different production stages. We saw Ascaris and Trichuris much more prevalent in the growers and finishers, and Ostracismum was more prevalent in the breeders. And so when we talk about the parasite load in different production stages on the infected farms, we saw with Ostracismum, statistically, there wasn't any difference among the production stages in the infected farms. We saw at 38 breeders, 42 growers, or 66 finishers that were infected with Ostracismum. However, with Ascaris and Trichuris, it was a little bit different. We saw, we had 8 farms that were infected with Ascaris, which is the roundworm, and we had by far more finishers that were infected with Ascaris, and then the second most heavily infected group were the growers, and the breeders were the least heavily infected. With Trichuris, there were 4 farms that were infected with Trichuris. Again, we saw more infection, almost equal amounts of growers and finishers compared to the breeders. Not nearly as many breeders were infected with Trichuris. And again, Trichuris was the whipworm. And so here we go. This shows just a little bit in a bar graph form what we were looking at with the parasite load at the different production stages. With Ostracismum, not much difference all the way across, but with Ascaris and Trichuris, we saw a lot more of it in the breeders and the growers. And so as we evaluate what we saw with prevalence of parasites, we saw that all forms were infected with at least one species. And across the farm, 72% of the pigs were infected with parasites. So again, it's something that we knew was out there. This was a way that we could quantify it, looking at different settings of organically raised pigs. And again, the heavier load of Ascaris in growing and finishing pigs was much heavier in those stages than they were in the breeders. So our second objective was to determine the effectiveness of manure composting. And this was to see if putting the manure that we collect in our housing settings into a compost pile or a compost row and getting it heated up, whether that had any effect at neutralizing or killing the parasites. So this is just a picture. This is Rick Carr from the Rodale Institute, and he's got he had set up the compost situation out at Rodale at Kutztown, Pennsylvania. And he had the equipment out there that he was able to make a really nice compost row, and then insert thermometers into the pile at different levels just to record the heat that was being produced within the compost pile. So our objective was to determine the effectiveness of composting manure on eliminating swine parasites and also its underlying mechanisms. And so we were trying to see whether the high temperatures that are generated during compost were enough to inactivate or destroy parasite eggs. And then we also were collecting samples before composting and then weekly for 4 weeks during composting. And so we set this project was set up. The grant was funded in August of 2018. So that was, late summer 2018. And the plan was to have a compost setting in the winter of 2019 and then in the summer of 2019, and to compare those two. And so this was done out at the Rodale Institute in Kutztown, Pennsylvania. And we conducted bioassays to determine whether the eggs were either inactivated or destroyed. And then we were assessing egg viability at different temperatures and durations. So the January 2019 composting results are shown here. And you can see that they were, the gray marks along here are the ambient temperature through January and February. And this was again winter of 2019, early 2019. Ambient temperature was around freezing. These temperatures are expressed here in degrees Celsius. And so at 0, that's freezing. And you can see that the red dots here, if you can see them, those are the times when the compost was turned. And Rick Carr had a compost turner that he hooked to the tractor and took it down and ran it along the row. And so it did a really great job the same way every time. And you can see also that the temperature of the pile was variable too. It kind of heated up at the beginning, but then it dropped off, and then there were a couple of points where it actually was warmer outside than it was in the pile. And this lower graph shows the associated eggs per gram. And what we're seeing here is that the neutralization of the eggs was very variable as well. And so, we really believe that was, the variations that we saw in the neutralization of the eggs were related to, excuse me, the variability of the heat in the pile as well. And so when we did it again in June and July, you can see that of course the temperatures were higher, the ambient temperature was higher, but also the temperature in the pile remained higher. And it wasn't just because the ambient temperature was higher, it had to do with we really had the management of the compost pile was less variable as well. But what we saw, and you can see, we've got heat and turning that's going on here. The heavy line indicates the temperature in the pile, and then the gray line is the ambient temperature, and you can see it was varying between 10 and 40 degrees Celsius. And you can see down in the lower graph, this is also the eggs per gram that we can see. And at the beginning, they were the different eggs are shown here, with Ascaris being the top one here, and the middle one is osphagastumum, and the bottom one is trichuris. And there's some variation in their livability, but almost immediately we see that the osphagastumum dies and is dead forevermore. Whereas, but we did see declines in the livability of the other two as well. And by week 7, we've got them all down pretty completely neutralized, or they've perished. And so that was kind of what we were hoping to see. And it was great to see that it actually came out that way too. So objective 3 was to assess bio-fumigation and see bio-fumigation as an approach to swine parasite control in pastures. And so this photo here is one of the bio-fumigation plots that was planted, and this was done out at the Rodale Institute as well. And this again is another view of the bio-fumigation plots that were planted. And so for those of you that are say, well, what in the world is bio-fumigation? Well, certain plants have an ability to, because of compounds located within the cells, they have an ability to suppress soil-borne pests or diseases. And so particularly plants in the mustard and brassica family, when they are crushed and broken, they release a compound called a glucosinolate. And when glucosinolates combine with enzymes and water, they're transformed into isothiocyanates. And isothiocyanate releases a gas that can be lethal to certain soil-borne pests and nematodes. So the thought here, and this has been bio-fumigation with using these plants has been studied in several different settings in the upper Midwest and across the country as an organic approach to control of nematodes and also certain soil-borne pathogens in crops. And so these plots of certain bio-fumigant plants were planted in May of 2019. They grow to a certain size, and generally right at or before flowering is when they need to be crushed. And so this is typically done with a flail mower, and the crushing of and grinding of the plant is what sets off the transformation and the production of isothiocyanates. And the other that needs to happen is when these plants are broken up, they need to be immediately incorporated into the soil for the gas that's produced to be to come in contact with the soil-borne pathogens. So these plants were planted in plots. They were artificially inoculated with fresh pig's feces. I mean, the manure was taken to these plots and spread on the plots, and then the plots were mowed with a flail mower in order to crush the plants and set off the transformation process. And then they were rototilled down to about 6 to 8 inches, and then they were rolled down too. And you can see here, that's what this is, that they were rolled with a cultipacker, and Rick Carr worked with several different types of management techniques to try to get enough pressure on the cultipacker in order to get to incorporate the crushed up plants thoroughly into the plot. And then they were sampled, the soil was sampled weekly for 4 weeks. And this actually was happening at the time that we were there. And so those results haven't yet been fully processed and released. And so we don't have particular data from that at this time. But that was the point of incorporating bio-fumigation, the bio-fumigation study into this project. The fourth objective of the project was to actually graze pigs in a pasture that had been planted with bio-fumigation plants. And that was to take place this past summer, growing season 2020. And with the advent of the COVID-19 pandemic, a lot of our work was curtailed. And so the bio-fumigation and the grazing results will come at some time, but they aren't available currently. So with that, I'm open now to any questions that anyone might have. Here is the details of the grant that we received. It was a USDA National Institute of Food and Agriculture, NIFA grant, and it was part of the Organic Transition Program of USDA. And again, it's a 3-year grant, and so we're continuing to gather the data and process it. But with that, I will stop my share.
Yeah.
And take it back to Olga.
Yes. Yes, we've gotten quite a few great questions, Diane. So
oh, excellent.
Yes, we have quite a few. So I'll kind of start to categorize them, but I'll just kind of also take them as they have come in.
Okay, perfect.
Are you using USDA Organic Survey for those percentages? And then it's an abbreviated IOW. How did you arrive at the 30% and 60%? Farrow to finish operations only, or did you also do feeder to finish operations? That was all one question in multiple questions.
Well, we did use feeder to finish operations in order to get in those data, if there were parasites present, all of the, well, as we had it separated by stage as well, but for that was all stages of pigs.
Okay. And does outdoor access mean that the pigs can go outside whenever they want? For the non-certified, what certified practices did they follow or not follow? And then IOW, at least cert organic feeds, grains, apples, ACV, whatever. Cert
right.
Organic straw bedding, any antibiotics? So it's kind of a big question.
Yeah, none of these used antibiotics, added hormones, or any synthetic dewormers. Let's see, the first part of that question was
outdoor access.
Outdoor access. Yes, they were free to go indoors and outdoors. Even the ones that weren't certified organic, they operated as you would consider the certified organic, because with certified organic, they are required to be able to go outside if they want to, every day of the year. And so these all had that option. Now the ones that farrowed in the winter, they were farrowing inside inside a barn, but they in the winter, but they farrowed out in pasture in the summertime. Now, what was the middle question there, Olga?
Yeah, and
The middle
it's
part.
for the non-certified, what certified practices did they follow or not follow?
Now, the non-certified producer in Wisconsin that had the Berkshires out in the pasture, and I mentioned that he fed apples and pumpkins in the fall. He also fed, he was non-certified, so I'm going to say I'm not exactly sure that he was feeding organic feed. The ones that were certified organic, they were using organic straw, organic wheat straw, and they were feeding organic feed.
Gotcha. Okay.
Yeah.
Okay. Did you look at breed differences in being parasite resistant?
We did not. It happened that most of the breeds we were looking at were hybrid. They were crossbreds of some kind, or Berkshire cross. We ran into a lot of Berkshire genetics in all of these farms. And you know, Berkshires have a reputation for producing a carcass and delivering meat that is more flavorful. But we didn't have that as one of the elements of this part of our study.
Okay. What, if any, non-synthetic dewormers were used in the study?
There were some, and if you're familiar with organic certification rules, there are some rules that some elements of it that a producer is allowed to deworm breeding stock prior to their third trimester of pregnancy, of gestation. So pregnant sows could be dewormed in the first or second trimester, but not after that. And so, you know, just sort of colloquially, that you almost think that could have had an effect of why we didn't see as much parasite load in the breeders as we did, although not all of the producers that we worked with actually were deworming their sow herds. However, they were using organically approved dewormers, and I don't have that right in front of me. I can't tell you exactly what those were. But again, not all of the breeding herds were deworming their sows, but I think there were 2 of them that we were working with in this of the 9 that had breeding herds that would deworm the sows prior to the third trimester of gestation.
Gotcha. Thank you.
Mm-hmm.
Is there a guide training available for me to determine my pasture parasite load?
Oh, that's a great question. And I think that there isn't. I don't think there is at this time. I know that Iowa State University has got some really good niche pork production information, but I'm not sure that would be part of it. This is something, you know, we're kind of taking this step by step because we don't have, there isn't a quantification through the universities or from research based. There are lots of good, there's a lot of good information out there. Of course, as part of extension and with university work, we'd like to be able to say that we've quantified this and that, you know, and so that there, you know, so that it has some gravity to it. And so we're doing it step by step, and this was really kind of just the beginning. First, Dr. Dr. Yushi Lee had done a survey the previous year. She had a smaller grant from USDA to just survey organic pig producers across the country just to find out how many there were. And then this was kind of the second step. How are we going to figure out how much parasite load there is? We know that it's there, but how much is there? And so that's the step here. And then the other three objectives were kind of to try to figure out if there were some ways of, incorporating compost into a way to neutralize the bedding and manure that we've got, and then also to look at bio-fumigation, which that's still happening, but it really got curtailed through the summer of 2020 because we weren't able to travel or work together with our collaborators in other states or, really not even across the state.
Yeah, seems like a great opportunity for sure to
Yes.
hopefully down the road provide some sort of guide for pork producers to be
Right.
able to, address that on their own farm.
Right. And that was part of the grant as well. And that's where I come in, because with extension, I'm about disseminating information to producers. And so, what we learn is going to help us be able to develop materials.
Yeah, perfect. What seasons did you gather fecal samples?
Well, we were gathering them in the summer season in June and July, because that's when they were outside on pasture. And of course, the composting that happened out at the Rodale Institute, those samples or that manure was actually collected from their pig barn there in the winter. And they have a really nice hoop barn situation there, and they actually cleaned it out. I think they cleaned it out 3 times a year. So we were looking at January, July, and November. We're going to be when we collected bedding and put it into the compost setting. But as far as the samples where the team went out and picked up feces in the pasture and in the bedding and the soil, we did that in the summertime, June and July.
Okay. Any advantages to rotating pigs, sheep on pastures?
Well, we haven't looked at that. We're sort of focused on pigs right now, but I would assume that could be something that we could look at in the future. At our research and outreach center in Morris, Minnesota, where Dr. Lee works, they have quite a bit of organic research going on. They've got an organic dairy herd there, and then they've got the organic and more natural or niche type pig production going on there too. And so that could be something that could be worked into, I think, some of the work that's being done at the West Central Research and Outreach Center.
Perfect. Was there a correlation with worms and pasture versus concrete?
Well, that's a great question. We didn't see that. Of course, it was summertime in all settings, because we did find parasites everywhere. That's a great question. We found parasites everywhere. We thought that maybe there was some warmth and cold weather effect on the Osteogostomum prevalence. We saw them more in the breeders, and we thought maybe there might have been, we might see more of them in the winter, but we weren't able to make collections in colder weather. We talked about it at the time, but at this point we haven't yet done that.
Gotcha. All right. Does apple cider vinegar help with parasite load? If so, when do you start adding it to the pig's diet, water? How do you add it, and what is the dosage?
Oh, that's a great question. We're familiar with the discussion of apple cider vinegar and parasites and other different kinds of management techniques. At this point, we haven't done any work with that in particular. We were just looking at these, at the bio-fumigation and the composting as ways of perhaps neutralizing parasites. But again, a great question, and just another indicator of how much interesting work there is to be done in this area of parasites in pigs that are raised outdoors.
Yeah, indeed.
Yes.
Three more. The next question is, has there been any study performed looking at soil types, mob or rational grazing, and or pasture plant types to help break down the presence of pasture parasites over time?
Well, that was, I'm kind of unfamiliar with all of the research that's being done. And this bio-fumigation grazing part of our project, which was actually being slated to happen this past summer, was our stab at it, because we wanted to see if actually running the pigs out in the pasture where they would be consuming and crushing up the bio-fumigation, the brassicas and the mustards, and we were using some clovers too. If that would be a way to neutralize the parasites or reduce the load, you know. At this time, we don't have data for that, but you know, that was our little stab at it. And I would think that you know, it certainly could be built upon to in future research.
Yes, yes. Since all three of these parasites are zoonotic, I'm not sure if I'm saying that, are there any thoughts of human studies to assess the risk to those that work with pigs that are not getting conventional anti-parasites?
That's an awesome question. That's an awesome question. And again, that wasn't anything that we were focusing on. But again, I think that it's sort of it's a natural lead in to talk about that too, because we're always concerned about biosecurity, no matter what the size of the operation is. And when you're dealing with zoonotic, whether it be diseases or parasites, again, making sure that we're protecting the team members that are working with the pigs is highly important. Great question. That is a great question.
Oh, there's more great questions
coming. Oh, good.
We have an engaging group here today.
Oh, good.
Yes. On the biofumigation, is it the green plant part or the fruits of the plant that need to be crushed?
Well, it's the green plant part, and I mentioned that the project that we did with Rodale out in Pennsylvania, the plots were planted in May, and they grew up. The mustards were actually just flowering, and so it was almost like, you know, when you're cutting hay, you want to do it just right at flowering time. You don't want to get too far into maturity, and that was the case with this too. That's when they were, so there weren't present. They were just beginning to flower when they were cut with a flail mower, and that really crushed them and broke them up. And that's the mechanism that starts the transformation of the with enzymes, water, and the glucosinolates in the plant parts. And then the incorporating of them into the soil immediately, so that the gas that is produced actually has contact with pathogens that are in the soil. And so again, it's kind of an early maturity sort of a thing when they are actually cut down and crushed.
Okay, this is another question on biofumigation. I'm assuming no financial analysis was done on the mowing, crushing, etc., versus a survivable worm load threshold. How can we check back in it to get the update on the biofumigation project?
Oh, yeah, we didn't, no, we weren't measuring that. And this was on a small scale too. Our plots were pretty, I won't say 20 by 20 maybe, but it was small scale. But of course, what we would want to do is be able to come up with a way to manage this that would be economically efficient. But our plan is to continue to disseminate this information, and you can as we work along and come up with some data and some results, these will be available through Dr. Lee at the West Central Research and Outreach Center with University of Minnesota, and also the Rodale Institute will be providing data too. You can go to their website and they talk about their pig production and the studies that they're doing as well. Again, we were really just rolling good when COVID hit last spring, because the sort of the rubber meets the road on bio-fumigation was going to happen last summer, and we weren't able to complete that then.
Understandable. COVID just threw everything.
Yeah, it sure did. Yes.
Enough for so many people, that's for sure. Were any of the producers using natural internal parasite controls on their sows boars?
I think they were. I think there were two. They were non-synthetic deworming methods. Two of the 9 farms, but I cannot tell you what they were. Unfortunately, I just don't know that.
Okay. I noticed that the compost temperature dropped sharply immediately following each turn. When turning the compost, does the heat loss or reduction reduce the chances of killing parasites in the compost?
Well, we think that probably there is some, when the temperature in the compost pile declines, the neutralization power declines as well. What we saw in the summer compost was, however, that the temperatures remained higher, more consistently high throughout the time of the compost. And it was 4 or 5 weeks that they were checking they actually had thermometers stuck into the pile with monitors. And so it was electronically recording the differences in temperature. And then the samples were taken out of compost and analyzed back at the lab at Kutztown University during each week to see whether the eggs were still viable, or how viable they were. But yes, I think probably when there's a reduction in temperature, the killing power is reduced. But what we saw was it always rebounded. The temperature always rebounded. And in order for the compost to actually be good to be used to spread on the fields as fertilizer later, it does need to be turned.
Okay, thank you. Were any of the pasture farms multi-species grazing? Did you notice any soil types that held onto the parasites more, clay versus sandy loam?
Excellent questions, both of those. The answer to the grazing was no. In these cases, they were all just swine graze. And I think maybe the farms were selected for that reason also, just to not confound this study. Let's see, remind me, the second part of the question was?
The second part was, did you notice any soil types that held onto the parasites more, clay versus sandy loam?
Now that one, I can't answer that one appropriately. I'm sorry to say, I just don't know.
Okay.
And I'm not sure if we were if we were keeping track of the soil type. I think maybe we were, but I can't speak to that one, unfortunately.
Okay. Parasites in organic swine and organic swine herd health is simply a very important topic that the organic pork industry really needs for future success of organic hog farmers and pork companies. I want to say thank you for all of your hard work. So that's not a question, more of a comment.
Oh, well, thank you very much. That really means a lot, because this is, you know, sometimes the organic or the niche or natural side of pork production gets sort of, you know, dismissed because it's not huge numbers, but it's important. And consumers today are interested in eating meat that's been raised organically. And heavens knows it's not easy to raise pigs organically. So I sure appreciate that comment. That gives us encouragement to charge on, because there is important work to be done here.
Indeed. Yes, I couldn't agree more. And I will piggyback on that comment too. And thank you as well from the bottom of our hearts for doing the work that you do, because it is very important. It helps so many of these smaller organic producers to know
That's
how
right.
to best, you know, treat their pigs. All right, the next question is, I am not familiar with hog operations. Are breeders kept indoors more, or are they at lower densities? What is different about their care that could explain the lower parasite load?
Oh, well, that's a good question. There are probably few of them. I mean, when you're looking at an operation that has a breeding herd, and they are also feeding growers and feeders, they would keep their growers and feeders probably in a more denser setting. However, I think that we may have touched on it a little bit earlier, that in organic swine, in organic certification, breeding herds are allowed to be dewormed at certain times, and it has to be within the first 2 trimesters of a sow's gestation. She can be dewormed, and so that may be another reason why we're not seeing as many, not seeing such a heavy load in the breeders, because they may have been dewormed at some point, and you know, that's sort of taken care of their issues for a time. It always will return, but that kind of keeps them down a little.
Yeah. Next question is, have you or your colleagues looked at older historical veterinarian information, meaning how to manage parasites when we didn't have modern tools, thinking now of indigenous knowledge?
Oh, well, we haven't gone back as far as indigenous knowledge, but I do have to show you. I've got this book right here in my little office called Swine, and it's and I had marked it because I kind of wanted, it was written in the early 1900s, and it was a University of Illinois professor, and he had written this book on swine. And at the time, swine were kind of a second thought. You know, people didn't really raise pigs, but he talked about some ways to control parasites that were, it's a wonder they didn't kill all the pigs, you know, kerosene oil and that kind of stuff. However, I haven't really looked back further than that. It was just, my brother found that book in a secondhand bookstore, and I've gotten a big kick out of reading it, because a lot of things hold true today, and a lot of things just, you know, we've gone beyond that, even in the management of organic and natural pigs on the small scale. But certainly that would merit some look too, just to see what indigenous practices might be related to management of livestock, of any kind of livestock.
Yeah, indeed. When was that book published, Diane?
It was, I think it was 1912.
1912. Okay, so about
Right.
a century ago then.
Yes, and it was a professor named William Dietrich, and he was at the University of Illinois, and he was also in charge of the university's research station at that time. And so I like history, and I kind of get a kick out of seeing how my predecessors worked, 100 years ago.
Yeah. Yes, indeed. The next question is there any research going on with regards to management in organic systems?
And other kinds of research going on?
The question was mange.
Oh, mange.
Yeah.
Yeah, that's another parasite that we deal with in outside hogs. I don't know that there's much I can't speak to mange research. That's one that, oh my, mange and lice, both, even when I was growing up and had a small pig herd for my 4-H project in the summer, we always fought lice. And so again, I'm running down another trail there, but I can't really speak to research that's being done as far as mange at this time.
Okay. Is there a herd size that would by itself minimize the parasite problem?
Oh, that's a good question. Probably there is, as far, because and it kind of depends not just on the herd size, it's more of a density thing. What is the facility size? If you've got 20 acres and you've got 10 pigs, you're going to see a lot less problem probably than if you've got 20 acres and you've got 40 sows and you're raising the offspring of that too. We haven't gone there yet. I mean, all these are great questions to prompt further study, as we go. But yeah, I can see that would be a great, I'm the kind of person that likes to be able to look up and say, yeah, if you're raising organics, if you've got organic sows on pasture, the stocking density is, X number of sows on 5 acres, and for worm control. So I think that probably is, we'll make a few notes and take it back to the team, because that would be very useful to know.
A great follow-up study.
Yes, it would sure be.
Yeah. And then there is more of a comment in regards to apple cider vinegar. I'll read it out loud for folks here.
Okay.
And the person is saying, I have done 3 replicated trials with and without apple cider vinegar and measured a benefit using apple cider vinegar, looking at internal parasites with supporting observations from our local vet service in cooperation with Practical Farmers of Iowa. A question was asked about dosage rate of apple cider vinegar, and the person is saying, I have always used 5 gallons per ton.
Oh, very good. That's good to know. And I know that there has been the Practical Farmers of Iowa, have done a lot of good work in livestock and crops both. And so thank you for that.
Organic question. Sows can be dewormed with synthetic dewormers every litter prior to last trimester. So she herself is never certified organic, but her offspring are. So she can be dewormed with chemicals twice a year?
Yes, prior to the 3rd trimester of her gestation. Yes.
So the mom, the sow, she's not organic certified, but her offspring will.
Her offspring would be, yes. I think that the sows that were in our study though were not dewormed with a synthetic.
They were dewormed with something that was approved by the
approved
National
for organic.
Okay.
Yeah.
Okay. That would make sense.
That I may be incorrect there, but I thought that was the case.
Okay. And Gary is typing into the chat box that the questions are done. So you guys, you obviously have the opportunity to ask more questions. Diane will be available for another, give or take 13 minutes. So definitely type in more questions if you have any more that have popped in to your head.
Well, that's great. Those are great questions.
Yes, there were, so many of them for sure.
Yeah.
Mm-hmm. So we can give it a couple more minutes to see if any other questions come in. If there, okay, here's another one that has come in, Diane.
Okay.
It says, as far as I last knew, or maybe it's a comment, ivomestrin, I'm not sure if I'm saying that right.
Ivermectin, yeah.
Was a sunset product last year, so sows are not allowed to be dewormed by synthetic
ivermectin
anymore at all. So it's more of a okay. That must be somebody that has organic,
that is certified organic.
Yes. Mm-hmm.
I try to keep abreast of that, but I can't really address that, but I'll take a look at that. Mm-hmm. I'm not surprised.
Mm-hmm. And then another question is, how do parasites affect health and production?
Oh, well, yes, the problem is, and it all depends on load and prevalence, but even the presence of a few parasites is going to diminish the productivity of the pig or of any animal that's got parasites. And so we try to manage our livestock, any livestock, but here we're talking about pigs, so that they're as healthy as possible. And parasites, there are a couple of ways that parasites can adversely affect. First of all, they are a parasite taking nutrients and causing the pig not to grow and gain as efficiently as it could, as healthily as it could. But also the presence of a lot of parasites can cause the pig to have sort of comorbidity problems where they can be more susceptible to other illnesses, not necessarily parasites, but a weaker, less healthy, or less thrifty pig has a better chance of getting sick if there's an influenza outbreak, or if there's a change in weather that might cause the pig just to not be able to bounce back as well. And so for that reason, that's why we want to try to reduce and eliminate the presence of parasites in a pig, just to keep him as healthy as possible.
So would you say that basically the parasites in pigs weakens their immune system in a sense?
Well, yes, it can. Yes, it would do that. And yeah, and so there's the direct parasitic effect that they're taking nutrients away from the pig and causing them not to grow as well, but also causing them to be be more susceptible because of their you said it, their immune system.
Yeah. And the other question, it says, you might have already mentioned this, but are you publishing results from these studies soon? I look forward to reading. This was a great session. Keep up all this needed research, and thank you.
Well, thank you. Thank you for a nice compliment. Yes, the plan is to publish these data. Unfortunately, our 4 objectives didn't all get completed in the time frame that we had hoped because of the pandemic. But yes, that's part of the requirements of the grant. And so we'll be doing more of these kinds of presentations as we have the opportunity and as we learn more and have more data to share. And then we'll also be publishing them. You'll be able to find them at on the Rodale Institute website or at University of Minnesota, either extension or Dr. Yushi Lee is at the West Central Research and Outreach Center. She's part of the animal science faculty with the university, but she's based out in Morris, Minnesota on the research farm. And so a lot of the research that happens out there, you can find that kind of research either on the university's website or animal science.
So do you have an anticipated date on when you think you'll be publishing the results of this, Diane?
Well, I don't know. I don't have a date to mention right now.
Okay. Okay, perfect. Well, I believe it looks like this is all the questions from what I can see, from what Gary tells me. Gary, thank you for sending me all the questions. And Diane, thank you for being here today, and thank you for the amazing.
Well, thank you.
This was just thank you for all the amazing research, and good luck with the rest, with the final piece of it. And hopefully
Yes.
you can get around to the farms a little bit easier this year. And
I hope so.
Yes.
Well,
so,
thanks so much. This has really been something. And on behalf of Dr. Yushi Lee and Dr. Alex Hernandez and Rick Carr and myself, thank you for letting us present this, our research thus far. And we look forward to connecting with our friends in the organic pig business in the future years too.
Yes. And hopefully more follow-up studies to come, right?
That's right.
Yeah.
Yes.
Awesome.
Thank you.
Well, and thank you everybody that attended today's session. We are so grateful you've taken the time out of your schedule to be here today. And I have a slide I just need to share with everyone. This is the slide.
There we go.
There you go.